Thank you so much. It's a pleasure to be here to present our developments on the high throughput screening microscopy. Actually, we are developing multisensorial hyperspectral and multimodal microscopes for identifying massive population of targets in microscopy. I'd like to, for those that are not familiar with the field of the high content or high throughput screening microscopy, this technology has played a very significant role over the years on molecular biology specifically, and it includes in a single platform imaging modalities, robotics, image acquisition, data handling, and so on, and has revolutionized several fields in molecular biology and towards developing new drugs and new diagnostic and treatment methods. In fact, this system is being able to automatically scan all kinds of molecular population or cellular populations, primarily being tagged with fluorophores. Sometimes they are using multiple fluorophores, and this way they, at the end of the analysis and the acquisition, there is a multi-step pipeline that includes the analysis of this massive amount of population of the data, and at the end of the day, it comes up with enumeration of the cells which are positive or negative, etc. And it has played, as I said, a significant role all this period of time. However, the main issue of this technology is that it primarily works with fluorescence sensors. So it has one sensor that captures fluorescence for 2 or 3 fluorophores, and these systems are not intended to provide real-time composition analysis in microscopic samples. It's just to identify fluorescent positive or negative biological specimens, to enumerate them and to classify in different categories. So we have capitalized over this, we try to capitalize over this significant technology because the way it works today, the analysis, I'll start from the field called forensics imaging. What they do, they actually collect traces from the crime fields, and the task is then to analyze which are these traces, whether it is semen or blood or DNA material or a variety of different kinds that can be found, either are human or natural materials. So the idea is it possible to scan a full region of a crime field and then put them on the microscope and do an analysis that runs in a completely unsupervised manner, and that after 3 or 4 hours to give you results about the chemical identity of the traces and to be able to classify whether there are existing some materials. Not only in forensics, though, there are many applications, industrial or scientific, that people want to do the analysis in an automated manner in microscopy to figure out where there are features of diagnostic importance and so on and so forth. So in order for a high content screening microscope to be able, within like a few seconds per field of view, to analyze and to identify the features that are seen under a microscopy objective, the way it works today is they're using several complementary techniques, including fluorescent microscopy, FTIR, Raman, spectroscopy, polarized microscopy, hyperspectral microscopy. And so there are many different devices, different sensors, that there are several problems in comparing results from the different sensors, and it is very time-consuming task, actually, in being able to pass a sample through all this pipeline of microscopes and spectroscopic techniques. And that is the problem that we want to address with a multisensorial high-content screening microscope that we have developed. And in fact, what we do, we have the ability we want to identify even traces or features that are transparent. There are many of those, like collagen, for example. Collagen is transparent, it's scattered, or some other traces in forensics are transparent. In this particular case, we emphasize in measuring and mapping anisotropy properties of the material, and particularly we talk about birefringence. Second, we want to identify the spectral characteristics of the materials, which are related with electronic structure and other structures of the specimen. So the multisensorial aspect of our system is that it can do snapshot real-time hyperspectral imaging with the devices that we have developed and represented in Outstand. We do multi-wavelength fluorescence excitation using multiple lasers for fluorescence excitation and multiple wavelengths for the imaging. And we also use polarizing microscopy, but the particular contribution is that we have developed a method to measure the birefringence in any pixel of the specimen. Birefringence measures actually is a measure of the some materials that are presenting optical anisotropy. They have two indexes of refraction, and this is, if you measure the difference of these two indexes of refraction, you have an index that can characterize a material based on the birefringence properties. And at the end of this acquisition process, we collect like 19 images from the hyperspectral, 9 images from the fluorescence, and 12 images from the polarization, which are modeled with Stokes polarimetry. And so we end up with a data set which is, includes all these images. And this is the block diagram of our system that has all the transmission, reflection, and polarization optics. This is the acquisition. So we acquire all these 40 images within 3 seconds. And then there's a training process with golden standard material to make chemical identity maps using machine learning technology. So this is how the system looks like. It has a large surface over which the user can dispose all kinds of samples, either there are industrial samples or there are biological samples or forensic samples. And then you leave the system to work without supervision. So what it does, it goes in its particular field of view and captures all these 40 images, fluorescence, hyperspectral, and birefringence. And so all these images provide complementary information for, it's very unusual for a material having, different materials have exactly the same characteristics in all these imaging modalities, which give us, as I said, complementary information. And at the end, we are generating a training data set where with ground truth material, these feature maps are correlated to particular chemical identity maps. This is an example. I'm going to end my presentation with 2 examples, one from forensics and one from histology. So this is from forensics. They are using tapes to collect the traces in the crime fields. And if you ask the forensics department to analyze, identify all these tapes, it will take like 2,000 years for them to analyze frame by frame. It's impossible. And that's why what they do sampling. So they select 2 or 3 sites that they, with the naked eye, select as the most probable to find some traces. And they do this analysis this way. So it's selective and subjective. So the new guidelines is that you need to tape all the crime field. And then there is a massive amount of background material which are not important, like soil from the ground, but also there are trace evidence materials that are very useful. So we have developed the system, as I said, and we have ranked it first in a European tender, and we have installed this system in the NFI in Netherlands, in the National Forensic Institute. We have installed this in the United Kingdom, in the DASA Center of the Defense Minister of UK, and also in Lithuania and other countries. And we have, as I said, many installations, and we received some Best Paper awards for this work. And the end point is actually, you see how an image looks, and this it's an original tape which includes hairs and all kinds of of traces, it's impossible to enumerate them. But here, in a completely unsupervised manner, it not only identifies here or fibers or things like that, but also enumerates them, which is very important to figure out what is the privilege. So the high content screening capacity here is actually demonstrated in this slide. We capture about, as an example, to give you an idea, is like 3,000 fields of view with an objective of ×5 or ×10. You generate 144 images with 3 or 5 seconds per field of view, that means that the acquisition time is 5 hours and the analysis time after the training is also 5 times, because 5 hours is very fast. If you do this same field analysis with like 1/3 of the techniques that the system integrates, it takes 37.5 days. So it's not possible to do this. So we improve the workflow by a factor of 180%. So that is for forensics, and this is becoming a standard now in forensics labs across Europe. And now that's another application where we exploit the birefringence aspect that we have developed. So what's the problem? I don't know if I'm sure very few people are familiar with amyloidosis. Amyloidosis is a very important disease which leads to dysfunction of several organs and also the failure of several organs like cardiac tissue. And today, the way that they diagnose amyloidosis is using a stain which is named as Cogored, and they use polarized microscope. But if you have, and what they try to do, the Cogored aligns with the fibers of collagen, and they change the birefringence of the material, so it's very characteristic. They give you some colors, interference colors, on polarization microscopy. The problem is, though, that these colors are very subjective, the estimation, and because the optical axis of these fibers can be in a random positioning, these colors change also because of the random positioning of the optical axis of the birefringence material. So that's a problem in finding a way to diagnose whether this is amyloidosis is positive or negative, and there is a fight internationally. When they see red colors is positive or green colors are positive, it's still ongoing, this fight in the histology journals. So we developed, our method is completely orientation independent, and intent up to this is maps where, as you see, this is the regular polarization image. There's not much information on the concentration of the amyloids, the amyloidosis, and then in this image, which you have a map of the birefringence, you see exactly the location and the problem related to the dysfunction of the conformation changes in collagen structure that it's induced because of amyloidosis. So what we're now going to do is that we introduce a new diagnostic index for histology, and this index will be birefringence, which is a physical property. And this will be completely dependent now from subjective estimations, and we hope that this way the impact will be huge, because amyloidosis affects hundreds of organs, and there is no trustable diagnostic process as of today. Coming up to the conclusions is that we have developed a multisensorial, multimodal, high-content screening microscope for quickening and automating the analysis in a variety of fields. We have a new method and technology to analyze in real-time optical anisotropy and integrate to the same system. We have a new technology for identifying and triaging in forensic traces, which is completely unsupervised and objective. And this is a ready product. We have already installations, but at the same time, it comprises a valuable platform, research platform for pharma industry and other industries, like anywhere you will need seed sorting, pharmaceutical products, molecular biology, film, polymer, etc. Thank you so much for your kind attention, and I'm happy to. I'm within the time, right?
Yes, exactly.
Excellent.
Thank you very much for this interesting talk. Are there any questions?